Today we will discuss about the process of glycolysis. And as we know, the glycolysis word is come from Greek word glykos. Glykos means the sugar and lysis means the breakdown. Therefore it stands that the breakdown of sugar. In this case the sugar is the glucose, and through enzyme catalyzed process the glucose is divided into two 3 carbon molecule of pyruvate, and during this serial enzyme catalyzed reaction, the that is the degradation of glucose, the energy is released from the glucose is highly conserved into the ATP and NADH. Next, this glycolysis process is occur in aerobic respirated organism as well as anaerobic respirated organism, and it is occurred in the cell cytoplasm in both type of organism. And this glycolysis process is invented by Meyerhoff, Emden and Chapman. This glycolysis process is that is the breakdown of glucose is occurred in 10 consecutive pathway that is catalyzed by the different enzyme, in which the 6 molecule glucose is divided into 2-3 molecule compound, that is the 2-3 molecule pyruvate, and these steps are divided into two phases. First is the preparatory phase, in which the glucose is converted into glyceraldehyde 3-phosphate, and this in which involve the 5 reaction. First reaction is the conversion of glucose into glucose 6-phosphate. This reaction is catalyzed by the hexokinase, and in which the ATP is harvested and generate ADP. And next, in second reaction, the glucose 6-phosphate is converted into the fructose 6-phosphate by phosphofructokinase enzyme. This is the second step. And third step is the fructose 6-phosphate is converted into 1,6-bis phosphate by the phosphofructokinase enzyme. In like the first step, this step also involve the harvesting of ATP, and which generate the ADP. And next, the fructose 1,6-bis phosphate is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by cleaving cleaving the fructose 6-phosphate, and this process is catalyzed by the aldolase enzyme. And next, the glyceraldehyde 3-phosphate is generated from the dihydroxyacetone phosphate by the triose phosphate isomerase enzyme. This process is the isomerization process, in which the intermolecular rearrangement is occurred. Among these first 5 reactions, the first reaction, that is conversion from glucose to glucose 6-phosphate and fructose 6-phosphate to fructose 1,6-bis phosphate, these two steps are irreversible, and other steps are reversible reaction. Next, we come to the payoff phase. This phase is involved the last 5 stages. In this step reaction, the glyceraldehyde 3-phosphate is converted into the pyruvate, which is both of which are 3 molecules. First step of the payoff phase is the conversion of the glyceraldehyde 3-phosphate into 1,3-bis phosphoglycerate. This enzyme is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, and in this process the NADH is generated from NAD plus, and one inorganic phosphate also harvested. And next, 1,3-bis phosphoglycerate is converted into the 3-phosphoglycerate by the by the phosphoglycerate kinase, and in this process the ATP is converted in from the ADP by adding of one phosphate. So this process is the generation of ATP. Next process, the 3-phosphoglycerate is converted into 2-phosphoglycerate by the isomerization process. This process is catalyzed by, that is that this reaction is catalyzed by the phosphoglycerate mutase enzyme. And next 2-phosphoglycerate is converted into the phosphoenol pyruvate by the enolase enzyme, and phosphoglycerate the phosphoenol pyruvate is converted into the 2 molecule pyruvate by pyruvate kinase. In last 5 stage of reaction of the payoff phase, only last reaction is irreversible. So in this process the glucose is 6 molecule, which is the 6 molecule, is converted into 2-3 molecule pyruvate. And this is the process of glycolysis. Hope all of you are understand. And this pyruvate will is used in later Krebs cycle or other process. Thank you.